In this video, we're going to look at the answers to the Unit 1 to 3 past paper retrieval questions. So there were 12 multiple choice, and then there were 43 marks of section B. Some of these were quite tricky, so we'll go through them. The graph below shows temperature changes during repeated cycles of PCR. If you start with 500 molecules of DNA and predict how many copies there will be after 20 minutes. So the first thing you need to do is think, well, PCR doubles the number of copies of the sequence that we have at each stage. In 20 minutes, how many cycles will I be able to do? So it's sometimes a bit tricky here to say, well, where does it start, where does it end? This is just basically like National 5 physics. If you pick a point on the graph and see how long it takes time-wise, so we're at 5 minutes here, how long before you get back to this part of the shape? So it looks like 4 minutes later at 9, you get back to the start of this bend, and you can do that with any part of the sequence. So it looks like after 4 minutes, we repeat the cycle. So in 20 minutes, you should be able to do this 5 times. You should have 5 rounds of PCR. So if we double the 500 5 times, so doubling it once gives us 1,000, then 2,000, 4,000, 8,000, 16,000. So the answer to the first one is A. This one here, we're looking at gel electrophoresis and paternity suits. This is a common problem-solving question. We know that each of these lines has to come from somewhere. So it either comes from the mother, in the case of this line and this line, or these two bands here don't seem to come from the mother, so they must come from the father. For the top band, we have the option of B and D. For the bottom band, we have the option of C and D. D is the only one that provides the two bands that are missing from the maternal strand. So D is the answer to this one. So this one here, sickle cell anemia, is a human condition affecting hemoglobin and the ability to carry oxygen. It's caused by a mutation that changes an adenine to a thymine, so we should know that's a substitution. It's one of our 3 single gene mutations: substitution, insertion, and deletion. So that is a substitution mutation, C. Also, it's part of the higher human course. The diagram below shows conversion of starch to maltose. So it looks like here, we're breaking bonds, releasing energy, and we're going from a large molecule down to many small molecules. That is a catabolic reaction, and it releases energy. So the answer is B. For number 5, we're looking at an experiment here. There's a snail in a glass tube. As it breathes in, the colour of liquid here rises, and then as it breathes out, the colour liquid would drop back to where it was before because of gas laws. However, when it breathes in, the liquid goes up. When it breathes out, the CO2 is absorbed by the solution here. So that means that the way we're measuring the metabolic rate of this snail is by its consumption of oxygen, because that's the direct measure we're using. So it's oxygen consumption, C. Which row in the table identifies components of the CNS and PNS? So this one's actually very tricky because one set of the pairs aren't the proper pairs. So brain and spinal cord, fine, those are the CNS, so it's A or B. But then you would expect parasympathetic and sympathetic and somatic and autonomic. Those are not some of the options. However, somatic and the medulla could be considered, obviously part of the autonomic. So I would say A is the answer to this one. Transfer of information from cerebral hemispheres occurs through the corpus callosum, so that allows the left and right brain to communicate and swap information. This one, again, some people find tricky because it's not obvious what the last little bit should be. But for 8, the following statements are events occurring at a synapse: an impulse being generated, acetylcholine diffusing across the cleft, being broken down, and being released. So if we think about it logically, we are trying to move an electrical impulse from before the synapse to after the synapse. So the thing that happens at the end is that this impulse is likely to be generated. That would be the logical thing to do, but we'll follow it through. So an impulse gets to the synapse, the acetylcholine is released from the storage vesicle. So we start with 4. So it's either A or C. Then the next option is the acetylcholine diffuses across the cleft, and we're then left with, does 3 or 1 come first? Now, logically, they happen more or less at the same time. But for the story in our head to work, once that acetylcholine diffuses across the cleft, it binds to a receptor and an impulse is generated. We then break down the acetylcholine using an enzyme. So we probably finish on B, because if you broke down the acetylcholine before the impulse was generated, it wouldn't be generating an impulse, basically. So that's the logical way to do it. The answer is B. So for this one, the function of a glial cell is production of myelin, B for 9. For 10, a drug trial was carried out. Again, this one, it's very easy to misread the units, as I did for about 5 minutes trying to work the answer out, and feel quite stupid at the end. So mice of an average body mass 20 grams were given 1 milligram per kilogram of their body mass of the drug. The drug was given twice a day for 15 days. What was the overall total mass of drug given to your average weighted mouse? So you've got to remember that 20 grams is not a kilogram. So how much of a kilogram is that? So if you take 20 grams and divide it by 1,000, you get the proportion of a kilogram. Because if the mouse weighed 1,000, you'd be dividing 1,000 by 1,000, and you would get 1. If the mouse weighed 500 grams, you'd be dividing 500 by 1,000, and you'd get 0.5. So that makes sense. If you weighed half a kilogram, the number 0.5 would come out. So it does work. So 20 divided by 1,000 will give you 0.05 possibly. You would then say, well, actually, that's being given twice a day. So you double that. And then it's going to be being given twice a day for 15 days. So you times that number by 15. And the number you get there is 0.60. If you treat it as a kilogram, you would get the number, what would you get? Something like 60. And then you would divide by 1,000 to turn kilograms into grams. Multiple sclerosis, MS, is an autoimmune disease in which lymphocytes destroy the myelin sheath. We talked about myelin before being produced by the glial cells. The row on the table showing the type of lymphocyte involved. So it's an autoimmune reaction. So allergy was B cells and autoimmune was T cells. And because you're destroying the myelin, that will decrease the speed of nerve transmission. So the answer is D. The table shows the number of cases of a disease, the number of deaths resulting in a year. You can ignore that because all it wants is the greatest percentage of deaths. So you would say 45 divided by 1,070 times 100, 12 divided by 420 times 100, 60 divided by 1,960 times 100, and 90 divided by 2,290. When you do that, I can't remember exactly, but you get a number, it's something like 5%, I think, is A, and that's the highest percentage. The rest are around 3, I think. This one here, protein X, you don't necessarily need to know this is a histone protein. It's one of the proteins that the DNA is wound around when it's not going through cell division. Explain why a primer is necessary for DNA replication. So the exact answer to this, because I might as well read that out, is that the primer is required for DNA polymerase to bind to. So DNA polymerase can add nucleotides to the 3' end of the new strand, or to give DNA polymerase a start point for replication. You can't say to start or initiate replication alone. You need to mention the relationship with DNA polymerase for this one. For C part 1, explain why the only leading strand can be replicated continuously. DNA polymerase, underlining the word polymerase, you need to say that, needs nucleotides. So it adds nucleotides to the 3' end of the primer or new strand, or DNA polymerase replicates in a 5 to 3' direction. It can only add to the 3' end, which is obscured in the lagging strand. So something along those lines. The enzyme joining the fragments together is ligase. We looked at some different mutated bacteria. Each has a mutation in a different region of its DNA, which is subscribed to RNA. We looked at different cultures of the mutated bacteria, calculate the simplest whole number ratio of the percentage protein in X, Y, and Z. So X is 9, so write 9 here for a second, or above, because it's not the final answer. So 9, 15, and 90. If you divide all those by 3, you get 3, 5, and 30, which is the lowest you can make that go. So 3, 5, and 30 is the final correct answer. Name the other component of ribosomes apart from RNA. This is one of those things. Like at National 5, you had to know that the cell membrane was phospholipids and proteins, but you didn't need to know anything else. RNA and protein is what makes the ribosome. You don't need to know anything else about rRNA. So rRNA and protein, so the answer is protein. Using your knowledge of biology suggests how changes in the sequence of bases in DNA transcribed to rRNA in the mutated cultures resulted in a decrease in protein synthesis. So the answer here is, here we go, see if I can just show you. A change in the sequence may change the structure, function, shape of the ribosome, or ribosome is not made, formed, usable, or translation can no longer occur. So you need both those. Or mRNA cannot join to ribosome, or tRNA cannot join to ribosome, to mRNA. So any of those is fine. It wants a structural difference between DNA and RNA. So DNA is double-stranded and RNA is single-stranded. DNA has thymine, RNA has uracil. DNA has deoxyribosugar, RNA has ribose. You have to do a comparison if you want the mark for that one. You can't just give one thing. You can't just say DNA is double-stranded, RNA isn't. You have to give the full comparison. For the next question, it's two choices of essays. So again, I might as well just show this. So for the first essay, you have 6 marks that you can access. You can get any 4 of them. So first mark, on the inner membrane or the cristae of the mitochondria. So if you mention that location, you get the option of a mark. NAD, NADH passes, carries the electrons and hydrogen ions to inner mitochondrial membrane or electron transport chain. The third mark, electrons are passed along the electron transport chain or along proteins or along a series of carriers. The fourth possible mark, energy pumps pass hydrogen ions across the inner mitochondrial membrane. Fifth possible mark, hydrogen ions flow back through ATP synthase. And the sixth possible mark, ATP synthase produces ATP. So you can access any 4 of those. Writing an account of competitive and feedback inhibition of enzymes. So the inhibitor binds to the active site is the first possible mark. Prevents substrate binding or joining, the second possible mark. Competitive inhibition can be reversed or overcome by increasing the substrate concentration. You could also mention the non-competitive can't, but that extra bit doesn't get you the mark. Feedback inhibition occurs when end product is at high or critical concentration. The end product inhibits, binds to the first enzyme or an earlier enzyme in the pathway. That's the 5th mark. 6th mark, this blocks the pathway or prevents synthesis of end product. So those are how you get those marks. The bond that holds the polypeptide chain in shape, so remember you've got your primary structure and then it folds by the hydrogen bonding, holding that polypeptide in shape. The gene for myoglobin consists of 1154 bases, while its mature mRNA consists of 462 bases. How come? So the answer to that is that introns or non-coding sequences are removed from the primary transcript. Or you could say that the gene, primary transcript, has introns and exons. The mature transcript consists only of exons. Or you could just say RNA splicing. It's best to try and give a reasoned explanation, but if you've said RNA splicing, you have the mark. For B, section of a chromosome or gene is added to its homologous pair. You have to mention that, the word homologous pair. Every time duplication comes up, the marking scheme is very strict about saying a gene moves from a chromosome to its homologous partner or homologous pair, or a section moves to the homologous pair. This is the tricky one, so it's good to practice these questions though. State the purpose of including a control group in the investigation. So this shows the effect of the mutations, or to show the effect of the drugs / each drug, or to provide the drugs effect increase, to prove the drugs effect increase chloride transport, or to compare with and without the drugs, not just the word to compare alone. So you have to talk about it being allowing a comparison between the drugs or the mutations or something like that. This one here, identify the treatment and mutation which has no effect. So you can see here with mutation B, no drugs and drug P have the same outcome. If they put a scale here, it would have been easier to see. But yeah, have the same outcome. So the answer is treatment P and mutation B. C, it says here an average is calculated, or the results have been averaged. So I don't know exactly where it says that. It does say that though. It says somewhere in here that the treatments have been averaged. Not 1000% sure, but that is the answer in the document. But anyway, we'll assume that it says it somewhere. So that's how you know that the treatment was carried out on more than one individual. Although this graph doesn't appear to have that on it, which is not useful. Anyway, give a role of the non-coding area of the genome. So the non-coding area of the genome, what it does is it can control transcription, it can transcribe to the tRNA or the rRNA, it can do lots of useful things. Alternative RNA splicing occurs. Explain how this results in different proteins being expressed from a single gene. So the result of a different combination of the exons formed in the mature transcript, or different exons are picked from the primary transcript to form the mature transcript. You can have either of those. In terms of activation energy, how do enzymes increase the rate of reaction? They reduce or lower the activation energy. It's quite a lot here. The exact location of glycolysis is just the cytoplasm, so you can just say cytoplasm. Describe the role of ATP in step 1. So it phosphorylates the glucose or intermediate, or it adds phosphates to the glucose or intermediates, or it gives energy to the glucose or intermediates. You can't just say it gives energy alone. You have to mention it to the glucose or to the intermediates. Explain how glycolysis results in a net gain of ATP. More ATP is made than is used, or more is gained. More is gained than used. Or you can say 2 ATP are used, but 4 are made. The actual numbers aren't really in the course description, but you can use them in the answer. So for the third one, removes hydrogen ions and electrons, passes them to NAD, or turns NAD into or to NADH. So that's how we do this conversion of intermediates to pyruvate. So when fermentation occurs, when fermentation occurs, state the conditions required for fermentation. It's an absence of oxygen. For the next one, using all the information given, suggests why the conversion of pyruvate to lactate is required for glycolysis. So it produces NAD to pick up more hydrogen ions and electrons, or produces NAD, which is needed for glycolysis. For the next one, looking at endorphins, we move away from these higher questions into the higher human questions. So state one activity that increases endorphin production. So we can have prolonged exercise, eating, sex, or laughter. You can't have stress or injury, but these don't negate a correct answer if you've also included them. You can accept any named sporting activity for exercise, accept any named food for the food bit, and any recreational drugs if you've mentioned one, like cocaine, alcohol, nicotine. The endorphins reduce pain or the intensity of pain. They induce a feeling of euphoria, pleasure, happiness. You can accept as a natural painkiller, or accept they inhibit pain or activates the reward path. But you can't say, sorry, activates the reward pathway, because then you'd be talking about dopamines. Parkinson's disease is associated with a loss of the dopamine-producing neurons. It cannot be treated by taking dopamine. Describe a possible mode of action a drug could be used to treat the disease. So the answer is it acts as a dopamine agonist. So it mimics the action of dopamine, which I always think is a bit unfair, because it's just specifically said if you give someone a chemical that is dopamine, then it doesn't work. So I don't really know why the marketing scheme suddenly thinks that some agonist is going to work, but that's neither here nor there. Anyway, it could prevent or slow the breakdown of dopamine, but you've lost the dopamine producing region of the brain. So again, I don't really know how that's going to work. Or it prevents, slows the reuptake of dopamine. Again, you've lost your dopamine producing neurons. So seems like, stopping the reuptake or destruction isn't really going to work. But anyway, who am I to question Mr. Forbes? Right, so question 9. The diagram represents a section of skin punctured by a splinter. Describe one way in which these epithelial cells defend against pathogens. So the epithelial cells form a physical barrier or are closely packed together. You can accept something like a waterproof barrier or producing sweat, sebum, or chemical secretions. You can't have stomach acid, saliva, mucus, tears because they've specified the skin. For the next one, structure X looks like a blood capillary. You can just say capillary if you want, though. And name the cells that release histamine. That is mast or mast cells if you want. For the second part, explain why the skin around the puncture wound will be red and swollen. So vasodilation or increased blood flow or increased permeability of the capillaries. You could accept a buildup of tissue fluid. Don't accept vasodilation of capillaries on its own. This doesn't negate if the answer includes correct reference to the capillary permeability. That's a bit unclear. But anyway, so vasodilation or increased blood flow or increased permeability of the capillaries. For C, it says, explain how a lymphocyte is able to recognize a particular pathogen. The answer is it recognizes an antigen. You need to mention the word antigen if you're going to go for that one, or antigens on the pathogen, or a specific antigen protein receptor binding site on the pathogen. So that was quite a lot of questions. Some of them are quite tricky, but I thought it was quite a good balance of problem solving, multiple choice, essay, and long answer question from across the course. Really, the function of these questions is to get you thinking about the topics you haven't looked at in a while. Make sure you have your summary notes and all your pupil notes up to date, and come along to any of the Google Meets, and either during or afterwards, ask questions about any part of the course. It's literally what I get paid to do. Literally get paid to answer your questions about biology. So it's definitely not an imposition because it's my job. So please come along, please ask questions. And if you have a specific past paper question that you can't do, bring it along and I'll look it up and we can go through it. So hopefully that helped.